Good afternoon, students. Good afternoon, everyone. Today I'll be starting about the aminoglycoside antibiotic. So, in previous classes, we have discussed about the beta-lactam antibiotics, and you know very well what is antibiotic and why we are starting about the antibiotic. So, according to classification, we have classified the antibiotics into different categories. So, antibiotic is one of the category. So, why it is saying as a aminoglycoside. So from the name you can know amino means the ring presence amino group, that is the NH3 and COOH, and glycoside, that is a link or that is the ring that is composed of a glucoside unit, and whereas 2 glucoside will be linked with a sugar moiety, that is known as the, and that compound is known as the aminoglycoside antibiotic. See the structure shows here. The structure is here. Now, aminoglycoside antibiotic is a special case of antibiotic where, coming to the key features, these are the branch of closely related chemically basic carbohydrate that are mostly water soluble, and the respective hydrochlorides and sulfates are crystalline in nature. Generally, the product will be in a hydrochloride or sulfated form. So they are found to be effective in inhibiting the growth of gram-positive as well as gram-negative bacteria. That means both gram-positive and gram-negative bacteria will both be killed with this type of antibiotic. They are also effective in the great extent against the different mycobacteria also. Relatively low margin of safety. They have the safety margin in low because they have some ototoxicity and a nephrotoxicity. So what do you mean by ototoxicity? That means continuous uses of aminoglycoside may cause hearing problem in the ER problem. And nephrotoxicity means they will be causing kidney problem. So whatever, without the minimizing dose, we can reduce that. Now coming to the preparation and mechanism of action. How it is prepared? Generally, they are prepared biosynthetically from an admixture of the streptomycin. So that is the admixture of the carbohydrate components of the fermentation media. Here is glucose units are fermented it, and different streptomyces or streptomycin is a drug that is derived from the streptomyces bacteria. So that is fermented. So mechanism of action, see, they usually act by causing the interference with the reading of the genetic code. That means whenever the genetic code, there is a DNA or the RNA code, that will be replicated, and in that case replication that will be prevented by this aminoglycoside antibiotic. So this is the gram negative bacteria cell wall, and this cell wall whenever created, because whenever any genetic material is formed, then they are trying to create a particular shell shape with the particular cell wall. So if the cell wall is prevented, then the growth of the bacteria will be prevented. So initially they penetrate the bacterial cell wall to reach the periplasmic space through the pore-in channels, that is the passive diffusion, and further transport across the cytoplasmic membrane takes place by active transport by proton pump and oxygen dependent process. Bind 30S ribosomal units and interfere the initiation complex. That means we know in case of the DNA or RNA transcription and translation, first initiation occurs. That means the starting of the DNA replication or the RNA transcription. So the 30S ribosomal plays an important role where they will be binding. As a result, the initiation or the starting of the DNA replication RNA transcription is inhibited. So induce misthreading of the genetic code on the mRNA and breakup of polysomes into monosomes. That means the mRNA will not be created, and as a result, the polymer of the DNA or RNA will be broken. As a result, the polysome will not be formed, monosome will be formed. Smaller fragment will be produced, so that will not be creating the DNA or RNA. Now coming to the glycosidic bond, see here. See glycosidic bond or O-glycoside. This is the anomeric carbon. This is the glucose unit present over here. See the glucose unit. This glucose unit, whenever will be reacted with methanol, so it will be producing a glycosidic bond, that is a OCH3, and that CH3 will obviously replaced with another ribose or adenine nucleotide. So we can see orthoglycoside and N-glycoside and S-glycoside here, that is required for our different synthesis of the drug. That is the orthoglycoside and N-glycoside, adenosine, ribose, and adenine. This is the adenine moiety, and this is actually present in the DNA or RNA. Now coming to the S-glycoside, see here where a sulfur containing a benzene ring will be produced. So basically this drug is generally composed of this type of orthoglycoside. We will be again going to the too much chemical action. See the structure of the elixir of the aminoglycoside. But before starting, we should know the aminoglycoside structure. It is an amino sugar. This product is a sugar, but whereas amino group is attached, that's why it is known as the amino sugar. Centrally placed hexose ring, that is the amino siloteacetyl ring. See, amino group 1, 2, 3, 4, and the fifth position there is the amino group, and there is a amino group. These two amino sugars are present. Sugar means dextrose means whenever they are attached with the amino group, they are known as the amino sugar, and that is attached with the ribose. See the ribose. So three amino sugar moiety are attached with the ribose group. So this structure is known as the aminoglycoside. So here R1 and R2 is mentioned. So in case of different drug, R1 and R2, the substitutes are changing. See, one of the amino sugar with the ortho group, the oxygen contains the amino sugar. Say here, this is also amino sugar. See, okay, two amino sugars are attached. And this B, that means neomycin. If this R1 is changed with the CH2N2, so this drug is known as the neomycin. And R2 here, H will be there, hydrogen. And for the neomycin C, the R1 will be hydrogen here, and the R2 will be the CH2NH2. Just alternate R1, R2. Two isomers are there. So, changing of this R1 and R2 group will leading to the different pharmacological action. Streptomycin, chiefly employed in the treatment of the tuberculosis in conjunction with other drugs such as isoniazid and rifampicin. That means this drug not only act in single dose, that's why they are required to combine with the isoniazid and rifampicin. Streptomycin and penicillin, synergistic action, subacute bacterial endocarditis, this type of infection will be inhibited or cured. Exert bacteriostatic action in low concentration and bactericidal in high concentration. That means the bacterial growth will be inhibited in low concentration, but in the higher concentration, the bacteria will be killed. In tularemia and bubonic plague, this drug is important drug of choice. So, brucellosis, this important drug, this is one of the infection is cured. So, for this detailed infection, you can go to the website also. So, for non-tuberculosis infection, usually 1 gram per day up to 5 to 10 days you have to consume. Now coming to the SR, detailed SR. The drugs serve as the triacidic base due to the presence of two characteristic chemical entities. See here, two characteristics entities here, that is a 2 amino sugar, and this is the 2 strong basic guanidinium moieties, and rather weakly basic methyl amino fraction. See this methyl amino moiety is present, that is producing the basic property. Now coming to the hydroxystreptomycin, that is the OH moiety present in this different amino sugar. If this OH moiety is replaced, one of the hydrogen atom of the streptose methyl function. So that means OH moiety will be replaced with the hydrogen atom of the streptose. This streptose ring where the hydroxy moiety will be removed, replacing one hydrogen atom. Streptomycin B, that is a mannose-streptomycin. So, mannose residue attached to a orthoglycosidic linkage. This mannose, this can be composed of mannose residue to a orthoglycosidic linkage via OH moiety at C4. If we nomenclature this 1, 2, 3, 4. what is C4 of the N-methyl N-glucosamine functional group that may act different way. This is a mano, so you can see residue attached to the orthoglycosidic residue via OH moiety at C4 of the N-methyl glucosamine functional group. Now coming to the neomycin drug. Used in a wide variety of local infections such as burns, ulcers, wound, impetigo, infected dermatose, paranculosis, that is or infection, conjunctivitis. Also employed as an adjuvant in topical steroid preparation to control secondary infection in case of inflammatory disorders. It displays broad spectrum activity against a good number of pathogenic organisms. It demonstrates a low incidence of toxic and hypersensitivity reaction. That means this is a more about safer drug. The drug is invariably combined with other antibiotics, namely gramicidine, polymyxin B sulfate, and bacitracin. Now coming to the SR of neomycin, the drug generally gets absorbed very rarely from the digestive system. Therefore, its oral administration primarily fails to produce any substantial systemic effect. So, as this drug is absorbed through orally and is rarely absorbed, so that's why oral dose will not be effective. Types: neomycin A, neomycin B, neomycin C. These are the another category of drug. So that neomine could be obtained by the methanolysis of the neomycin B and C respectively. That means neomycin B and C, these are the different component where it will be prepared by the methanolysis of this drug. Whereby the glycosidic linkage existing between the D-ribose and deoxystreptamine undergoes cessation. Kanamycin. It is also effective against some mycoplasma and gram-positive bacteria, for instance, S. pyogenes, S. epidermidis. Along with penicillin, it is found to be effective against Streptomyces faecalis. It is used alone or in combination for a variety of disorder: acute staphylococcal infection, gonorrhea, tuberculosis, acute urinary tract infection, for bowel sterilization in hepatic coma, and also prior to bowel surgery. This drug could not be useful in tuberculosis, perhaps due to the fact that it developed resistance to microorganisms rather rapidly. This drug is not applicable in tuberculosis also. Dose: oral adult for intestinal infection 1 gram after every 8 hours for 5 to 7 days, it will be effective. For preparative preparation 1 gram every hour for 4 doses, followed by 1 gram every 6 hours for 36 to 72 hours. So this is the SR of kanamycin that is also present over here. That is not required too much. So remember, there are some points. Kanamycin develops cross resistance overwhelmingly in the tuberculosis bacilli, especially along with some other medicinal entities such as vichromycin, dihydrostreptomycin, and antitubercular. The drug causes either retarded or impaired loss of hearing, ototoxicity. Kanamycin and penicillin salts must be combined in the same solution, perhaps due to the possible inactivation of the either agent significantly. So thank you everyone. Thank you for this study. Thanks a lot.